Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about the next part of the types of lipids from the paper Lipid Metabolism. The objectives of the schedule is to continue to know about the complexity of the lipids, the types of complex lipid present in the living organism. What are the significance of the glycolipids and how they act in a system? Actually, in last module, we have discussed about the glycerol-based glycolipids. In this module, to carry further, we will be discussing about numerous other glycolipids which is a part of complex lipids. So, we are discussing the different types of lipids, and these types include the simple lipids, complex lipid, and derived lipids. The simple lipids, including the fats, the oils, and the waxes, have already been explained in the previous modules. In the complex lipids, which includes the phospholipids, glycolipids, and proteolipids, has been discussed herein. Phospholipids are phosphorus-containing lipids, whereas glycolipids are lipids containing saccharides or the carbohydrates. When lipids are amalgamated with proteins, it forms a complex mixture of lipids which is called as proteolipids. So in this module we are discussing about different types of glycolipids, and it has been continued from the previous modules. In mycobacteria, the lipopolysaccharide, the phosphatidyl inositol dimannoside, lipoarabinomannan, and lipomannan has been the anchoring domain and holds the key significance in the interaction between the host and the pathogen. This phosphatidyl inositol glycans demonstrates the earliest version from the prokaryotes that are branded for the cell surface protein anchoring and the structural features of lipoarabinomannan have been used across the globe as the tuberculosis vaccine. The surface of the leishmania Entamoeba and Crithidia species contains Lipophosphoglycan. This lipophosphoglycan is a glycolipid, consists of 4 domains. The first domain is an alkyl lysophosphatidyllyl inositol. The second domain is the core of glycan. The third domain is the repeated units of galactose and mannose phosphate backbone. And the last or the fourth domain is the cap structure of an oligosaccharide has been involved in numerous functions inside the mammalian host. In the strains of Streptococcus species, a glycosylated cardiolipin named Glucopyranosyl cardiolipin was noticed, and it has been found that it constitutes about 18% of the lipid phosphorus. But present as a trifling constituent in Vagococcus fluviatilis species. In Deinococcus radiodurans, 3-galactopyranosyl 2,3-phosphatidyl glycerol derivative, which is an exclusive glycophospholipid of a fatty alkyl amine, has also been identified. This glycophospholipid contains about 33% of the total lipids. As shown in this structure, the R3 position, where an alkyl amine is present, are generally straight chain compounds. And these straight chain compounds is either C15 or C17 carbon units, or having C16 to C18 units, with isomeric 17:1 basis preponderant. Another subtype of glycolipids, which is glycosylated cardiolipin or glucopyranosyl cardiolipin, has been reported from the Mycoplasma fermentans, and this phosphocholine containing glycoglycerolipid where the phosphocholine group is attached to the carbon 6 of glucose moiety. This lipid valor in the pathogenesis of diseases like rheumatoid arthritis by eliciting inflammation or cell death by their phosphocholine residues. In Lactococcus and Streptococcus, which are gram-positive bacteria, a number of complex glycolipids deriving from glucopyranosyl 1,2-glucopyranoside diacylglycerol were recognized. Glucopyranosyl cardiolipin or glycosylated cardiolipins is another subtype of the complex glycolipids. In Staphylococcus membrane, additional complex phosphorylated compounds like lipotic acid, which are consist of the polymer of glycerol 1-phosphate, has been found linked to a glycosyl diglyceride or a phosphatidyl glycosyl diglyceride, which is present. The other substitutions on the glycerophosphate units with alanine or glycosyl groups have also been reported. The structure shown here of lipotic acid were depicted from the bacterium Bacillus subtilis, and if we look at this structure, the R1 as well as R2 positions are having fatty acyl groups, and the R3 position contains hydrogen. Alanine or acetyl glucosamyl group. Sulfoglycoglycerolipids are the lipids which demineur a sulfur atom found in an acidic membrane with a very strong or very acidic pH. Say for example pH 2. These type of lipids are present in mammalian cells, the chloroplast of the higher plants, some bacteria, algae, and also in fungi. sulfoglycoglycerolipids has also been described in Elecatothrix and Zygnema, the green alga, and in Fucus and Pelvaceae species. At a very low pH, the carboxylic acid occur in an unionized form, but sulfolipids subsist in the anionic form. In Chlorella species, a new type of sulfolipid, which is called as sulfoquinovosyl diacylglycerol, or abbreviately known as SQDG, is derived from monoglycerodiacylglycerol and was discovered in 1959 at the Scripps Institute of Oceanography, which is located in San Diego, United States. And these SQDGs was afterward described in the thylakoid membranes of all higher plants as well as lower plants. Hence, the sulfolipids are present in algae, cyanobacteria, few Basidiomycetes fungi, for example Clitocybe, Coprinus and Saliota species, and the photosynthetic membrane of the plants. The biological function and the mechanism of SQDG has been described in higher plants. Predominantly under the phosphate ravenous circumstances. The SQDG is the lipid with a sulfonic acid link containing monoglycerol diacylglycerol at C6 position of the monosaccharide unit. This sulfoquinovosyl, which is also called as quinovose 6-deoxydeglucose, is the sulfonoglucosidic moiety, 6-deoxy 6-sulfonoglucoside, where R1 is often the palmitic acid and R2 is the unsaturated chain. Sulfate esters of galactoglycerolipids are reported in a few mammalian tissues like brain, testis, spermatozoa. These constitute about 3% of total lipid and more than 90% of total glycolipids. The acyl group at R' and R" are most frequently saturated. That is, they contain either 16 or 18 carbon atoms. Seminolipids, which is a mixture of alkylacyl and diacylglycerol is associated with the myelination process of the nervous system. These seminolipids are also essential for the spermatogenesis as revealed in the studies carried out in mice. From Hyphomonas janasiana, which is a seawater bacterium, a tauroglycolipid has been isolated, lacking the phospholipid content. The structure determined as 1,2-diacyl 3 Glucopyranosyl glycerol taurinamide with 16 or 18 and 19 as major acyl chain. Among the ceramide-based glycolipids, out of the 150 known varieties in this cluster of glycosphingolipids, 50 of them are characterized into the structure of ganglion. They are strewn mostly at the surface of the cell and participate in the regulation of cell interaction and serve as a distinctive marker for cells, along with mediation of cell-to-cell detection as well as contact. These ceramide-based glycolipids are indispensable for the growth as well as development of an organisms and allude to the viral or microbial infections and cancer as well. They form molecular complexes named glycosides and helps in explicit recognition between the cell's function as well as signaling process. They are found in animals, in bacteria, in fungi, and in plants. The grouping of the glycosphingolipids is based on the structure of the carbohydrate moiety and is described herein. The first in this category are neutral glycosphingolipids, which is an unsubstituted glycosyl group. It includes single or extra glycosyl moieties, hexoses, sometimes pentoses, and an added hexosamine are present. And as per the presence of the glycosyl unit, there may be one glycosyl unit which is called as monoglycosyl ceramides, or there may be more than one glycosyl unit which is referred to as an oligoglycosylceramides, or other structure containing only one glycosylated sphingobase, which is called a deacylated glycosphingolipid, or one fatty acid which is linked to cerebrosides, which is acylated to a glycosphingolipid. So, complex sphingolipid with a ceramide containing a cyclopropane chain and prenalated residues on galactose, which is referred to as an plakocytes. The next category in this is acidic glycosphingolipids. They are the zwitterionic molecules with glycosyl and carboxyl group. Sulfate or phosphate residues are present, and the examples include gangliosides, which contains a carboxyl group on sialic acid, and several glycosyl residues. Other examples include acidic monoglycosylceramides, phosphonoglycosphingolipids, sulfosphingolipids, which are ceramide di and trihexosyl sulfate with one sulfate molecule on the glycosyl unit, which is called as sulfatides, and phosphosphingolipids, which are phosphoryl ceramide with oligosaccharide or glycosyl head moiety. Lipopolysaccharides are the endotoxins emanated from the cell wall of gram-negative bacteria. It is also found in a fungus named Antrodia camphorata. Due to their nature, they cause numerous pathophysiological symptoms like blood pressure decrease, diarrhea, fever, septic shock, and death. There are three parts in lipopolysaccharide. The part 1 or the part A contains the lipid A that seal out a composite with part B having a polysaccharide core through a glycosidic linkage, and this glycosidic linkage is allied to an external area of part C, which is an immunogenic and capricious polysaccharide chain or antigen composed of repeating oligosaccharide units that is liable for the specificity of the bacterial serological strain. The part A or the lipid A includes a backbone of beta-1,6 glucosylamine glucosamine with 2-phosphoester moiety at position 1 of glucosamine 1 and in position 4 of glucosamine 2. The position 3 of glucosamine 2 assembles the glycosidic linkage, which is an acid labile, to the polysaccharide chain. In Escherichia coli, a gram-negative bacterium, the supplementary groups are replaced with fatty acid as hydroxymyristate with 2 amide and 2 ester linkage, and laurate as a fatty acid. In the same species, the lipopolysaccharide fatty acid profiles vary distinctively from the phospholipids. The sterol-based glycolipids occurs as free sterols, the acylated form or the ester form, or the alkylated form or the alkyl ethers, sulfated form, or linked to a sterile glycoside moiety. The biosynthesis of sterol is ever present between the eukaryotes but almost wholly deficient in prokaryotes. Being absent in an archaea, they are sparingly distributed in bacteria and Methylcoccus capsulatus. For example, Proteobacterium. Gemata, Obscuri globus, along with Myxobacteria members. As a consequence, the existence of varied strains is primordial. Rox is used as proof for eukaryotic evolution some 2.7 billion years ago. The sterols have hoponides as precursors and an oxygen-dependent biosynthesis, starting with the creation of 2,3-oxidosqualene, which is a primary intermediate. Steroid and triterpenoid hydrocarbon detection in the history of Earth has been used to surmise the relic of oxygenic photosynthesis. It has been conjectured that the levels of oxygen increased in the atmosphere made the sterols evolution feasible and make possible the eukaryotes' birth, giving them with an untimely protection mechanism against oxygen. Among the sterol-based glycolipids, free sterols or the sterols shape an imperative cluster on steroids. The unsaturated involve a 3-beta hydroxyl group, carbon atom of an aliphatic side chain at position 17 figures the group of sterols. Being saponification resistant, they are endowed in all animal and vegetal tissues. Important in the appraisal of marine sediment, maturity as a biological compound. They also comprise 3-hydroxysteroid crystalline alcohols and are classed as triterpenes directly by unsaturation, cyclization, and 3-beta hydroxylation. cycloartenols in plants or lanosterol in animals. So, today in this module, or what we called as lecture, we have learned about the different types of glycolipids, the complexity of the glycolipids, as well as the different types, the presence of the different types in different types of organisms, ranging from bacteria to animals and to plants. Certainly, there are certain advantages as well as benefits of these glycolipids, and the presence of these glycolipids in the membranes or in the different types of organism itself explains the complexity of the evolution of different types of lipids. So this type will continue in the couple of forthcoming modules, and in that we will discuss in details about other types of lipids. Thank you.